This conference will now be recorded.
Okay. So first things first, exams, I went back over them on Saturday morning. Grades are already pretty good once everything got cleared up in terms of weird question translations to Canvas. Grades are even better. So I think the average was 87, 88, something like that. So that's pretty good. Pretty happy with that. Looks like folks are finding a groove, at least with the lecture exam, which is always a good thing. Where we have to go today in terms of, and again, as always, if you still have further questions about scores, you can always email me. I know some people emailed me over the weekend about stuff that was an issue with the Canvas quiz. But if you have other questions, of course, feel free to email me about that. What we have to do starting today is kind of continue on with where we were before the exam. And what this really is kind of a bridge system between the circulatory system that we just talked about and the immune system, which is where we're going next. The immune system, of course, is more than tangentially related to the circulatory system because a large part of the immune system involves lymphocytes, right? So that's going to be obviously a part of that. Here with the lymphatic system, what we've got is a very, very simple circulatory network in some ways that has been kind of co-opted or modified over time to also play a role in the immune system. So what it originally was, evolutionarily speaking, was a means of getting fluid back into the blood. So if we think about what's happening at a capillary bed, right? We have blood flowing through some very porous blood vessels very, very slowly. We are exchanging things with the interstitial fluid around us, right? But as we're doing this, we're also going to be leaking fluid out of the blood. We're going to be actively losing plasma. Okay? And as we lose plasma, right, we lose blood volume. And a big part of where we left ourselves last time was that the idea of losing blood volume is a profoundly bad thing. Okay? So we have to have a way of getting that fluid back into the blood. That's where the lymphatic system comes in. And so we're going to have the sort of the ends of the lymphatic system in and around capillary beds. So that fluid can be picked right back up, right? And we're going to eventually kind of converge back at the sort of the major veins of the body, dumping that fluid back into the blood before we get back to the heart. And that was originally the whole purpose of this thing. As we have kind of evolved through time, us animals in general, because of the ability of the lymphatic system to pick up larger structures, larger objects, whole cells, if you will, it was very, very useful for us as a means of surveillance in terms of immune surveillance, looking for bacteria, virally infected things, cancers, whatever. And so we started to see modifications of that system, the building of things like lymph nodes, of lymphatic tissue in general, like malt, to kind of house immune cells as part of that immune system. And so we have not just these lymphatic vessels themselves, but also things like lymph nodes. One kind of important distinction to make is that the fluid that we're talking about here is the same fluid that we call plasma. It's the same fluid that we call interstitial fluid. Okay, we just call it lymph when it's in the lymphatic system, just as we call it plasma when it's in the circulatory system. It's not any special fluid, it's just what we call it while it's in there. Additionally here, we have a number of different lymphatic organs that we'll talk about. Some scattered lymphatic tissue, so not necessarily part of a specific organ, but within or around other organs. And again, this is where those lymphocytes are going to be. We're going to house, we're going to store. Some of our lymphocytes are actually going to grow up or mature in parts of this lymphatic system. And so this would be things like the spleen. Spleen's a big one. The thymus, the tonsils, again, another really important set. And then stuff like mucosa-associated lymphatic tissue. And we'll talk about all of these in turn. We'll spend some time on all of these different organs to a greater or lesser extent. Okay. So we'll start though with the sort of the initial function here. What is this stuff originally there to do, right? Be a place where fluid can be pulled back in or returned to blood. And so the lymphatics are going to be the part that's responsible for that. And that moves a lot of fluid every day, right? We transmit about 3 liters of fluid from the interstitial, from the capillary beds, back to the blood every single day. And if you think about it, right, you have about 5 1/4 liters of blood in your body, which means that in the absence of a lymphatic system, you would lose more than half of your blood volume simply to leakage out of capillary networks every single day. You would survive less than 1 day alive, right? So it's a really, really, really massively important system for us for that reason, if nothing else. Okay? And in addition to just returning the fluid, returning the lymph, plasma, or whatever you want to refer to it as, it's also going to be required to pick up some of the bigger things that are there as well. So plasma, proteins, and obviously, we use it for things like, whole cells and stuff like that too. So the structure of these lymphatics nearby the capillaries, while the vessels themselves are very, very small, they're going to be built slightly differently to accommodate these larger structures that we have to kind of permit into them. So if we think about what this looks like for us, in kind of a systemic context, we have a very, very simple schematic of our circulatory system, right? This system, systemic circulation here. Heart, arteries coming out to a capillary bed, returning to the blood via the venous, or returning to the heart via the venous return system. Right? The lymphatic system is going to be this kind of little bridge here between those two systems. Okay, so we've got lots and lots of little vessels here, lymphatics down here in the capillary bed that, just like veins, join together to form larger vessels, that join together to form larger vessels, and eventually come all the way back to places like the subclavian vein, things like that, on the way back to the heart. If we look a little more closely here, in terms of the organization of these lymphatics, we have the largest ones we'll call ducts, and then trunks, and then from in there we've got nodes within the sort of the trunk system here, and eventually coming down to collecting vessels, and all the way down to the smallest lymphatics here, which are the capillaries. And that's where we're going to start looking at first. So again, here the idea is that these lymphatic capillaries are intertwined with all of our little capillary beds, right? And the way that they're going to be built is with this series of sort of one-way flaps or one-way valves in here. And we'll take a little closer look at this in a minute. Again, like the circulatory system itself, it's unidirectional. It flows only one way. Like the venous return system, it flows only towards the heart. Right? And again, it's structurally quite similar, right? Small vessels become bigger vessels, become bigger vessels. And so we think about that kind of as progressing from capillaries to collecting, trunks, ducts. With the lymphatic system, there are fewer named vessels that we have to worry about. Okay? Really, we're only going to be talking about naming the trunks and the ducts, because those are pretty consistent person to person. The remainder of the lymphatic system is highly variable between people. Okay, so we don't really get into the names there as much as we do. So starting all the way down here, the very, very smallest of these vessels, right? These little, tiny little capillaries, right? They're not dissimilar from blood capillaries at all. They're small in diameter. They're thin-walled, low-pressure kinds of things. What makes them special compared to, say, a blood capillary, again, is that sort of overlapping flap-like structure of the epithelial parts of the vessels. They're very, very, very permeable, right? They're more like, say, a fenestrated or even a sinusoid capillary in terms of what they'll be able to take up, right? In terms of size of things that they'll take up. So proteins are no problem. Cells, pathogens, cancer cells, all of these things can enter these capillaries. So they're capable of bringing in big stuff. That's half of the purpose here. And they do so, and we'll talk a little more about these in a second, because of these sort of mini valve structures that we've got on there. Now, what this means for us in practical terms is that not only can we pick up dead or dying cells, right, but also a lot of pathogens are going to be picked up by the system as well. So cells that we want to get rid of, right? And so again, when we talk about kind of co-opting or taking the system over and kind of using it for two purposes, right? Sort of adding in the immune function here. This is a big part of why. Right? This is kind of the easiest place for us to get it, because like the venous return system, we kind of take all of these different parts of the body and we connect them all to more or less one or two just very common places. Right? All of the lymph flows through a handful of vessels, a handful of places. So you can surveil the entire body, you can look at the entire body without having to have structures everywhere in the body, because all of the lymph, all of these pathogens, all of this stuff comes back to one or two centralized locations. Makes it very, very useful for us. As far as where to find these, they're everywhere that we've got capillaries, right? So places that are avascular, we don't have them. So they're not in your teeth. There are none in your bones, which is kind of surprising given the amount of vascularization of your bones, blood vessels in your bones. We don't have any lymphatics in there. We don't have them in cartilage, obviously, because in the blood vessels and cartilage, you don't have them in the cornea. We don't have them in the central nervous system, okay? For reasons that, if you recall kind of the organization of the central nervous system, may be obvious, right? We don't really have the way that we get blood and nutrients and things to the central nervous system is through the production of cerebrospinal fluid, which has its own circulatory network. So we don't have lymphatics. We return to the blood through sinuses, things like that. So we don't need it there. We also have a specialized set of these lymphatic capillaries associated with the digestive system, predominantly in the upper parts of the small intestine. And these are called lacteals. And the purpose of the lacteal is to take up fats. Now, we'll talk more about this when we talk about nutrition, so the last part of the course. But the way that we take up fats, because they're not water soluble, we have to actually kind of encase them in proteins. And so we end up making this kind of large molecule called a chylomicron out of a fatty acid and a sort of a capsule of protein. It's a big molecule. And something that large is hard to absorb through endocytosis, through any other means, right? And so the easiest way for us to do this is to dump it into something that is going to enter the blood anyway, right? But also is already capable of bringing in very, very large things, like a lymphatic capillary. So these lacteals are specialized specifically for the absorption of fats from the digestive system. So looking at how these things work, basically what we've got, okay, are all these kind of slightly overlapping cells here, right? And one end of these cells is anchored, and one end of these cells is kind of free. So if we take one of these cells right here, one of these endothelial cells, this end right here, we've got these collagen fibers that are anchoring us to the surrounding tissue. So this one doesn't move. But if we go up here and we were to push down on this, right, we were to push in, it would flap in like a door. There's nothing anchoring this side, right, holding it against this tissue from being pushed in. So big things can push this cell in and enter the capillary. But because these cells overlap, right? Because this cell underlaps this cell above it. We can't push out. We push out, we push against the cell next to us. It's how like a regular one-way door works, right? You can open a door one direction, right? But if you try to pull it open the other way, right, it's going to hit the jamb and you won't be able to come in. You won't be able to pull it. Right? In this case, we're going to hit the overlap with the other cell and we're not going to be able to come push out. So things can enter, but they cannot leave. So we have this one-way system. Again, like we have in all the other valves of all of our other associated vessels, right? Keeping things flowing in only one direction is very, very important for us. Okay, so these capillaries are going to coalesce, they're going to merge, they're going to get into fewer but larger vessels, okay? And at that point, we enter kind of the realm of the collecting vessel. The collecting vessel, you know, like we talk about veins, right? We talk about small veins and large veins, but we don't really distinguish between them in any real meaningful way. And this is kind of what collecting vessels are. They're a wide range of sizes. They are, you know, they merge to form other collecting vessels. They're just kind of the sort of the small vein, I guess, analog of the lymphatic system. They are, if anything, flimsier than actual veins. They typically have thinner walls. The fluid in here is under even less pressure, if you can imagine that. It's essentially under zero pressure. So we have many, many, many more valves, okay? Again, just to kind of keep stuff flowing in one direction, not flowing backward. And again, because, you know, we see in veins, right, we see all these anastomoses because we're under low pressure. We need to have detours. Here, again, zero pressure, so lots and lots of anastomoses with regard to collecting vessels. Really, again, trying to do everything that they possibly can to make up for what is essentially no internal pressure. They're going to travel alongside vessels. So we've got some that will travel alongside arteries relatively deep, some that will travel alongside veins relatively shallowly. But they kind of travel in those same, that sort of same company. As they come together, I guess before we get to that, so just like we had with large blood vessels, right, we had to have a special circulatory system just for the blood vessels, that vasa vasorum. In a large enough collecting vessel, we're going to have the same thing. Okay, again, because these are living cells, they have to have a blood supply. This is how we get to them. Now, as these collecting vessels come together, eventually they reach a sort of a large enough point at which we're going to refer to them then as a lymphatic trunk. And the lymphatic trunks are going to be named because they are consistent from person to person in ways that collecting vessels are not. And most of them are going to be paired right and left, but there is one that is all by its lonesome. So there are right and left lumbar trunks. There are, which are, of course, the lumbar region, right? There are right and left bronchomediastinal trunks, okay, which occur in the thoracic cavity in the mediastinum. Right? We have right and left subclavian trunks, which occur below the clavicle, like where the subclavian artery, subclavian vein are. And paired jugular trunks, so it's like the jugular veins draining the head. We have jugular trunks draining the lymph from the head. And then the one that is all by its lonesome is the singular intestinal trunk. And we'll see a picture of these all in a minute here. And these are going to come together into one of two ducts. Okay. We have the right lymphatic duct. The right lymphatic duct. So the thing with the ducts that's kind of interesting here is they're not symmetrical. One of the ducts drains almost the entire body, and the other duct really only drains part of it. So the right one drains only just a tiny little portion of it. Okay. The right lymphatic duct does the right arm and the right side of the head and chest. So if you imagine kind of a little rectangle here, a little triangle here, this is what that right duct does. The thoracic duct does everything else. left side of the body, its entirety, the right abdomen, the right leg, all of that stuff. And both of these, where they're going to come in, right, if they're going to come in essentially to the same place, they're going to return to the blood at essentially the same place, which is where the internal jugular and subclavian veins come together. So right at the transition point between those veins and what we're going to refer to then, of course, as the, oh great, I'm blanking on it. Oh my God, seriously. This is embarrassing. I can't remember the name of this term. Oh God. That's unbelievably embarrassing. I can't remember that. That sort of main vein that comes in there right before we return to the superior vena cava here. Brachiocephalic, that's what it is, the brachiocephalic trunk. So right where the brachiocephalic trunk begins, that's where these are going to come together. So if we look at a map here of the body, all of this here in this sort of sickly green color is what's drained by the right lymphatic duct. Okay, so you can see here again, just the right side of the head, the right arm, and the right chest, and everything else is drained instead by that thoracic duct. Thoracic duct runs here, okay. The right lymphatic duct here. If we look a little more closely at what's going on here with most of the organs removed, here we have the right and left, or right and left rather, lumbar trunks together here, the intestinal trunk in the middle between those, right? And these three come together to essentially form the thoracic duct. Now there is this little structure here called the cisterna chyli. I'm not going to ask you to remember that. It's just a swelling at the base of the thoracic duct where those three trunks come together, the lumbar and the intestinal trunks come together. The subclavian trunk, coming in from the left side here and the right side, jugular trunk coming up from the head, and the bronchomediastinal trunk coming down here. Now again, we got this sort of substantial issue, right? That we have no pressure. It's very similar to the same condition we had in the venous return system, right? Where we have to have some means of returning that fluid back under what is essentially no other impetus, right? And so we utilize a lot of the same things that we do in the venous return system. So we have skeletal milking, right? So we have that same kind of issue where we have skeletal muscles are going to be compressing these vessels and putting pressure on the lymph within them, moving valve by valve by valve by valve by valve. Okay, again, that's sort of salmon ladder kind of analogy that we had going on before, where we were kind of step by step moving back towards the heart. We have that respiratory pump we talked about before there also, right? So as fluids enter the abdominal cavity, right, due to the actions of the diaphragm during respiration, we push down and we compress the abdominal cavity against the pelvic floor. We increase internal pressure here. It's going to force that fluid, blood in the veins, lymph in the lymph vessels, doesn't matter. We're going to push that up into the chest. Lots and lots and lots of valves. Okay, again, like we said, even more valves than we're expecting to find in veins. We can, for the deep lymphatics, we can rely on pulse pressure. Okay, so just like we see compression by muscles, compression by a pulsing artery can sometimes provide enough impetus to move us along a little bit. It's not going to be a lot, right, but it is still there. And vasoconstriction, again, like with veins, something we can do. It's not really the most powerful means of moving lymph along, but in the absence of anything else, it will do it. Now, within the lymphatics themselves, of course, we also, the term here, lymphoid, right, lymphatic, lymphocyte, all kind of reminiscent of one another. And so within here we have, of course, a lot of those lymphocytes themselves, both B cells and T cells. When we talk about lymphocytes, they are going to be active in what we're going to call the adaptive immune system. So as we talk about the immune system, we have a couple of different parts of it. The adaptive immune system is the part that is responsive to specific pathogens. So when you get a vaccine, for example, like a non-mRNA vaccine especially, but even something like the COVID-19 vaccine, these are the parts that are going to respond specifically to those pathogens. The reason you have to get a flu shot every year is because those pathogens cycle every year. And so the adaptive immune system is great, but only in a very, very specific window of things. Okay? And that's primarily built on these T cells and B cells. T cells and B cells are looking for antigens. And we've talked a little bit already about the idea of antigens. We talked about blood typing, right? And the idea that if you get the wrong blood type, right, you have an immune reaction against it, okay? Because we have the wrong name tag. Right? You had an A protein when you weren't supposed to have one, right, or a B protein when you weren't supposed to have one. Those are antigens. Those are things that your active immune system looks for, your adaptive immune system looks for. Okay? Those things that say where you are or where you belong or who you belong to, that kind of stuff. And so when we talk about being active against specific cells, right, every strain of the flu, for example, modifies your DNA in a very specific way and modifies it for a very specific antigen. They will only really ever operate against things that they're already sort of predisposed to do. And so this can be anything like a foreign cell itself. So a bacterium in your body will have an antigen that you will not recognize unless you attack. Virally infected cells will modify the normal antigens on your cells because they modify the DNA in your own cell. Again, a mismatched transfusion, a cancer cell, very much like a virally infected cell, in that you have a modification of the DNA, okay, which very often results in a modification of the antigen on the cell as well. So the immune system here has I guess, kind of a way of recognizing a lot of potentially different problematic cells. Now what we're going to do here with these two different kinds of cells is kind of come at the problem from two different angles. So a T cell, generally speaking, there are lots of different kinds of T cells, and as we get into the immune system, we'll talk more about those. But generally speaking, when we think about T cells in the immune response, we're talking about cells, we call them cytotoxic T cells, that are actually actively involved in the destruction of particularly virally infected cells or bacterially infected cells. Something with a wrong antigen, an antigen that's not recognized. So playing a very, very proactive role, I guess, in the immune response. B cells, on the other hand, B cells are going to be a little more, be basically one step removed from that process. What they do is they secrete antibodies. And what an antibody does is it simply attaches to a cell, right, that has the particular antigen on it, and it marks it for destruction. Right? It's a way of labeling which cells have to go. Now, T cells don't attack antibody labeled cells. They don't. T cells and B cells don't work together. They have two very different responses. They're kind of held separately from one another, but they both work in kind of in the same circles, so to speak. Outside of lymphocytes themselves, we have other lymphoid cells here of use for us. So other leukocytes here, for example, things like macrophages, okay, are also involved in here. Now, a macrophage will attack a antibody labeled cell, okay, but only antibody labeled cells. We also have dendritic cells and reticular cells, and we'll come back and we'll spend a fair amount of time talking about dendritic cells They are surprisingly very, very important, particularly for the adaptive immune system. And then reticular cells here are going to basically kind of be structural. They're going to create what we call a stroma. We're going to come back and use that term stroma again and again and again. Basically what it is sort of like a loose netting, right? So we think about, for example, in the spleen, the stroma there is kind of a sort of a gentle hammock, right? But one that will capture and help eliminate broken down red blood cells, for example. So if we take a look here at the inside of a lymph node, we see something like this. So the stroma, right, is kind of the background structure that we see. So the scaffolding within this lymph node. Okay? And that's made out of this reticular tissue, reticular fibers. And within here, we have all kinds of different lymphocytes, right? So B cells here. Here we have a different leukocyte, a macrophage. Okay? And then throughout here too, we would also find that we don't see any on here, but we'd also see dendritic cells. Now, what does lymphoid tissue itself do? It is, again, primarily a place for this subset of immune system cells. In particular for lymphocytes, in particular for B lymphocytes. We will find T lymphocytes in there as well, but not sort of housed permanently or semi-permanently. And like we said before, one of the big things with the lymphatic system that makes it useful for this kind of purpose is that, again, it routes lymph from all over the body into one of two trunks, right? Or one of two ducts, right? One of 9 trunks, right? Just a handful of places, right, that we have to look. And by looking in just those handful of places, because all of it passes through those same doors, we don't have to look anymore. Right? It's an easy place for us to kind of easily surveil the entire body. Right? So it's kind of a logical place for us to kind of have all that. Lymphatic tissue itself is, again, mostly that stroma, right? It's mostly reticular tissue. We're going to be dealing with large chunks of things. We're going to be dealing with cells, and we want to have kind of a loose netting available to that. And so we can capture things so that we can gently kind of handle cells that we don't want to destroy, all that kind of stuff. Throughout the body, we're going to divide this lymphatic tissue basically into 2 different morphs, 2 different forms. Okay, we're going to have diffuse lymphatic tissue, which is exactly what it sounds like. It's kind of loose and spread out. Okay, or we're going to have dense lymphatic tissue in the form of a follicle, okay, or a lymphoid follicle. This diffuse lymphoid tissue is in almost every body organ, give or take. Okay? Obviously, places where we don't have lymphatics, we don't find it. So we don't find it in your bones, for example, but it's in basically everywhere else. And mostly where we find it are in mucous membranes, right? Which is where the term MALT, or mucosa-associated lymphatic tissue, comes from. And the reason that we have it in mucous membranes, right, if you recall, a mucous membrane lines a body cavity that's open to the outside world. Okay? And so if we're thinking about how are pathogens going to get inside of our body, that's a really good way for them to get in, right? Getting into these mucous membranes. So into your mouth, or the opposite direction, into your eyes, nasal cavity, right? Easy places for things to get in. And so if we're thinking about, well, where are we going to start looking for pathogens? The best place to start looking for them is right at the front door. Right? So we're going to have a lot of this lymphoid tissue there for that particular reason. Where we're going to find dense lymphoid tissue, though, these follicles, are primarily in lymph nodes. Okay? We're also going to have some of this dense lymphatic tissue or follicles in structures that we call Peyer's patches at the tail end of the small intestine, again, for reasons we'll talk about in a little bit. what this dense stuff is, what these follicles are, right, are places where B cells can grow and mature. So if we're thinking about what B cells are doing, right, they're encountering pathogens, they're generating antibodies against it. Okay. So again, thinking about what are we doing, what's the immune function of the lymphatics here, right? Surveillance. We're going to have a widespread body-wide immune response. A nice convenient place for us to have that, right, is with B cell generating all of these antibodies that will circulate in the blood across the entire body, right? And will eventually get to where they need to go. So we have follicles there primarily for that purpose. We also have a number of these in the appendix as well. So lymph nodes, Peyer's patches in the appendix. Okay. Lymph nodes themselves, when we think about the lymphatic system, we think about lymphoid organs. Lymph nodes are probably one of the first places to look. They're in clusters scattered throughout the body. That, not necessarily everywhere, but we do have a fairly widespread distribution of them. And like arteries, they only come close to the surface in a couple of places. So we've got them in your armpit, right? So we have the axillary lymph nodes, right? We have them in the inguinal region. So the inguinal region, if you don't know, is basically where your inner thigh and your lower abdomen meet. There's a passageway through there that the femoral artery passes out of. This is where, if you're a male, this is where the spermatic cord comes through there. Basically, it's just kind of in that region. And then in the cervical region up here at the angle of your jaw. And if you've ever gone to the doctor and been sick, you're probably familiar with one of the primary means of looking at infection without doing blood tests, right? Just palpate these lymph nodes. Usually here at the angle of the jaw is the one that we feel for, right? Because these will swell in response to an infection. Primarily because this is where B cells will proliferate, right? So as we start having an immune response, those B cells will swell those lymph nodes out, okay? And we'll kind of be able to feel them. Bubonic plague, so named because one of its effects was to generate these large swellings or buboes in the axillary region. So somebody with bubonic plague would be plagued by big black swellings in their armpits. Bubo for little owl. I don't know particularly why they use the term little owl for these things, but they do. And of course, also you can palpate these here at the inguinal region here. So axillary, cervical, inguinal. As far as what they're doing, right? Again, like everything else, right? We're trying to monitor with the content of this lymph. We're trying to clean out stuff before it gets back to the blood. And again, it doesn't have to be pathogens, right? It can just be dead or dying cells, or chunks of cells, or debris of any sort. Just anything we don't want circulating around the blood. Lymph nodes are really a good place for us to do that because of the way that they're structured. And the way that they're structured, pretty quick here, the way that they're built, right, is designed to slow the flow of fluid through them. Right? So there's all of these entrances, right? All of these little entrances here. And obviously this is a cartoon, but this is the idea. And just one or two places where they're going to lead. It's kind of like my favorite ride at Disney World. And I haven't been there since the Star Wars thing, so maybe that my favorite ride is going to change. But my favorite ride at Disney World is the Haunted Mansion. Right? You get in the Haunted Mansion, and it's kind of this tricky thing because they have all of these doors that you come in. And you think, the first thing I've ever been, I think, oh shit, this is going to be a fast ride because people are just moving into this ride at a fast clip, way faster than anything else. But it's a trick because you get in there and you get into this big atrium, and then you stop. Because there is only like one door out of this atrium onto the damn ride. And so while people can pile in fairly fast, because there's only one way out, you slow down and you're stuck in this room for a really long time. This is the same thing with how a lymph node works. all of this flow of lymph in, but only a couple ways out, means that lymph stays in a lymph node for a long time, right? It gives it a lot of opportunity to be kind of picked over and cleaned out, right? Lots of time for us to find anything in it that's got to go. So this structure here, this way that this is built, is really, really fundamental to that. Most of them are going to be vaguely kidney bean shaped, like we saw before. They're going to have, and this is going to be something that's going to carry over to most lymphatic organs. They're going to have an outer fibrous capsule that subdivides the inside into separate little rooms. So we have trabeculae that make us into little rooms. And there are two parts. There's an outer part, which is the cortex. The inner part is the medulla. Show you the picture here again. So here we have our capsule on the outside, right, with these little trabeculae that come in, divide us into little rooms. We have a cortex out here and a looser medulla in the middle. The cortex is where we find most of the follicles. The B cells are in the cortex, the outer part of it. There are going to be some T cells in a lymph node, but they're not there to stay. They're just there because they're traveling around the body. Okay, so when we think about lymph nodes, we think about follicles, then we're B cells. Here's an actual histoslide of one. Here's a fibrous capsule, trabeculae, all these little follicles in here, full of B cells. And here's that sort of looser medullary tissue there. Now again, the big deal here is the flow. Right? The idea that we get a lot of flow in, but very low rate of flow out. We're not going to be zooming through here. And again, this is just there so that the B cells can encounter anything. If there's stuff that needs to be broken down, we have time for macrophages in the medulla to do it. Just so we've got time. That's kind of the whole point. Last thing we'll cover today here, excuse me. The next sort of major lymphatic organ here is the spleen. Spleen embryologically is a site of blood cell production. The spleen and the liver take over from the yolk sac before being superseded by the bone marrow. But in the adult, at least the sort of post-embryonic world, it's there primarily to remove old red blood cells from circulation. So again, they last maybe 4 months, right, before they start to break down, they get brittle. And so really this is kind of the major point for us. We have a very, very, very large artery coming in here, the splenic artery, one of those branches off of that celiac trunk. The reason it's such a large artery serving it, because this is an organ of filtration, right? It needs to continually sort of cycle out the blood. Remember, we're talking about millions of red blood cells being removed from circulation every second. We have to have a lot of flow into the organ that's doing it. And if we look at it, I'm going to sit right here. So in this particular example, the stomach has been moved. It's been reflected out of the way. The greater curvature of the stomach would sit right here. The fundus would sit right here. So the stomach kind of sits like this. So it's tucked away kind of behind the stomach and to the side. Here's that big old massive splenic artery. It's hard to get a sense for how big this is from here. It's about the size of my pinky. It's quite a massive one. And again, right, we're breaking down red blood cells. We're storing the products of that breakdown. So we're storing iron. We're shuffling heme off the liver to become bile pigment anyway. It's a storage facility for platelets. Like I said, early on, it's where we make red blood cells before we have marrow. Primarily reticular tissue on the inside. They have very sort of expansive stroma within the important organ, but that's really all there is to say about it. So, okay, so I will see you guys for a lecture on Wednesday. We will finish off the lymphatic system and move on to the immune system. Have a good day, and I'll see y'all in